Hello everyone, I'm Dr. Vibha Khanna. In this video, we are going to learn about the regulation of gene expression in eukaryotes. Now, as is well known that regulation of gene expression is basically a process in which the external or the internal signals they control the response of the cell. These genes they are present within the nucleus in the eukaryotic cell. That is the characteristic feature of a eukaryotic cell, that is presence of membrane-bound organelles. The second characteristic feature is the presence of chromosomes, that is the nucleic acids. They are packaged in the form of chromatin network. They have got histone proteins associated with them, and these are the basic factors which help us, or rather which help the regulation of gene expression at various points. As we have already talked about in our previous videos on regulation of gene expression in prokaryotes, the genes, there are some which are expressed continuously, others are expressed during differentiation, and many as a result of cell differentiation. So this gene regulation, it can be of two types: the negative gene regulation and the positive gene regulation. Negative gene regulation refers to the regulation in which a repressor protein is involved, and it is this repressor protein which dictates whether the gene under consideration will transcribe or they won't. While the positive gene regulation, the molecule, the regulatory protein or the signal, they interact with the DNA and they regulate the expression of the gene. Now we have already done the lacoperon and the tripoperon. Both of them, they are examples of the negative gene regulation, because the regulatory protein basically represses the transcription of the polycistronic units in both the operons. The difference, however, is that the lacoperon is the inducible gene regulation, or it is the inducible type of operon, because in it this molecule, that is the allolactose, it deactivates repression. It induces the transcription process by removing this regulatory protein from this operator site, whereas the tripoperon is an example of repressible type of operon, in which the tryptophan, when in association with the regulatory protein, it induces repression of this polycistronic unit. However, while talking about catabolite repression, we have talked about this cap protein, which is activated by the cyclic AMP. Now, this is the example of positive gene regulation. It induces transcription of these genes of this polycistrone. So this is the example of positive gene regulation. In eukaryotes, mostly positive gene regulation is met along with. Now, in eukaryotic cell, since the genetic material is bound within and organelle, most of it within the nucleus. So the control points of gene expression, they are quite different and complex as compared to the gene regulation within a prokaryotic cell. So the first point is the chromatin structure, because in eukaryotic cells the genetic material is associated with some basic proteins, the histones, and hence they are not available for transcription. So by making them available or unavailable for transcription, this gene expression can be controlled. Now, once made available, the next step is initiation of the process of transcription. So, in eukaryotic cells, this initiation of transcription itself is a complex process involving a wide range of proteins. So again, this is the second control point of which we will soon talk about. Now, third thing is that once after transcription, the RNA is formed. Now, in eukaryotes, this RNA is the heterogeneous RNA involving both the coding and the non-coding sequences, the exons and the introns. So what we intend to is that all the coding sequences, they together form the messenger RNA or the RNA transcript that will be translated into the protein later on in the cytoplasm. So by controlling this RNA splicing, by controlling this RNA processing of this messenger RNA part, again the gene expression can be regulated. Now many of the genes, they form special structures like the RNA hairpin like structures form. Now if this hairpin loops they are produced, this RNA is not available for translation, or there are certain special type of RNAs like the small interfering RNAs or the microRNAs present within the cytoplasm, which form complementary base pairs with the messenger RNA, and thus it is made unavailable for translation. So this type of gene silencing is also made use of to regulate gene expression in eukaryotic cells. Next point of control may be the protein synthesis, that is the translation part, because translation itself is a complex process, and if all the component proteins, the basic essential required proteins, they are not made available, then these genes, they won't be able to express themselves in the form of proteins, the structural or the functional proteins. And once the amino acid, the polypeptide chain is produced, it is not that it will form a functional protein, because it undergoes many post-translation modifications, structural as well as chemical modifications. So again, at this point too, the gene expression, it can be stalled in a eukaryotic. Now, if we'll talk about these points of regulation of gene expressions, so first we will talk about the epigenetic regulation of gene expression. That is the mechanism that switches genes on or off. As we all know that the nucleic acid, the DNA material in a eukaryotic cell has to be compacted within the confines of the nucleus in the form of chromosomes, and this is done by organization and packaging of genetic material, and certain basic proteins, the histones, they play a crucial role in this process. First, the DNA double strand, it gets associated with the histone proteins. Nucleosomes are produced, then coiling and supercoiling takes place, and ultimately we get this chromosome structure. So, in order for transcription to take place, the DNA strand has to be made available for transcription. So, this can be done by modifying the histone proteins. Now, one thing that is to be noted in this figure is that these histone proteins, especially the H3 and H4 histones, they have got these N-terminal histone tails. And basically, the modifications associated with epigenetic regulation of gene expression, that is the methylation and acetylation, deacetylation, they takes place on this N terminal of the histone tail. So first we are going to talk about the histone acetylation and deacetylation process. This histone acetylation, it occurs at the lysine residue within this N-terminal tail of H3 and H4 histone proteins. And this acetyl group is provided by the acetyl coenzyme A, and the enzyme that catalyzes this process are the HATs, or the histone acetyl transferase. Now, this removal of this acetyl group, once this acetyl group is attached to the N-terminal tail, then this chromatin, it is made available for transcription. In order to remove this, now, since if we, in case we do not require any more activity of the gene concerned, so what happens is another enzyme, that is HDAX, or the histone deacetylases, they remove this acetyl group hydrolytically, and thus again compact packing takes place, and the genes, they are silenced, or transcription, it does not take place anymore. The second modification that plays an essential role in epigenetic regulation of gene expression is the methylation and demethylation of histone. Now, there's one thing typical about this chemical modification, and that is that methylation can activate as well as deactivate transcription. That is, it can repress transcription or it can induce transcription depending upon the residue that gets methylated, the amino acid within the histone that gets methylated. So the methylation is done by this enzyme histone methyl transferase, or the HMTs, and it is obtained from this S-adenosyl L-methionine and is transferred to the lysine or arginine residues of the histone proteins. So in the cell nucleus, when histone methylation occurs, there are specific genes which may be activated, and at the same time there may be other genes which on methylation gets silenced, and demethylation exerts the reverse effect. So this is the second type of modification. That now, after the DNA strand is made available for transcription, second thing is initiation of transcription of the gene. Now, In this figure, we are considering a gene X. Upstream this gene is the promoter region, and this promoter region has got two domains. One is the regulator domain, and the other domain is at which the RNA polymerase it binds. That is the binding domain. So over here, there are many proteins that are that play essential role for this RNA polymerase to bind to this binding site of the promoter and then initiate this process of transcription. So the first requirement is the general transcription factors. Transcription factors, they are the proteins which recognize specific sequences. They are induced by the environmental signals, and once activated, these transcriptional factors or these proteins, they recognize the specific site within the genome and binds to it. This signals the binding of the specific RNA polymerase 2 at this promoter of the gene that will be induced to express itself or induced to transcribe. This process, that is formation of this complex and then initiation of transcription, it has got the involvement of other proteins also. Other proteins which may be present upstream or in some cases even downstream, that is within the genome, they may be present anywhere. They are trans-acting regulatory proteins. So wherever they are synthesized, they are going to reach the specific transcription factors, or if they are the cis-acting units, in that case the part of the nucleotide sequence, the part of the chromatin, it will come in close proximity with this gene that is to be transcribed. So these are the regulatory proteins which may be activators or enhancers, or they may be the silencers. So as we were just talking about, these are the enhancers. They are present far away on some other part of the chromosome, and they come in contact with the transcription factors. Certain mediator proteins, they help in association of these activators to the transcription factors, and for the process some DNA bending proteins, they have got their own role to play, so that this distal control elements or these control elements which are not in close proximity to the promoter site, they come and lie near this promoter site. If activators are binded over here to the transcription factors, the rate of transcription it increases. However, if silencers are there in place of these activators, the activity of this gene or the transcription activity, it will be down regulated. So the transcription factors, the enhancers, the silencers, these type of proteins, they play a crucial role in regulation of gene expression in eukaryotic cells. Now, one more characteristic feature of eukaryotic gene is that in eukaryotes they are the split genes. By saying split genes, what we mean is that the entire transcript does not act as a messenger RNA. There are certain non-coding sequences which are not the part of the mature messenger RNA, which interrupt the coding sequences. These introns, they are the part of the nucleotide sequence which will be left behind within the nucleus after splicing, and only these exon parts, they will join together and form the RNA, the messenger RNA that will be translated in the cytoplasm, that will leave the nucleus, the nuclear membrane through the nuclear pores. So, if transcription of this interrupted gene, or what we call the split gene of a eukaryote, takes place, an entire transcript is produced, having both exons and introns transcribed. This RNA is the heterogeneous RNA, and it undergoes processing before forming the, so that it may form the messenger RNA. The three basic steps of processing: first is removal of these introns so that only exons they are there. Then capping, and in order to protect the 5 prime end of the messenger RNA of the RNA, and then tailing. Now, the length of the tail, the poly A tail that is added on this region of the messenger RNA. This poly A tail, the length of this poly A tail determines the life span of the messenger RNA within the cytoplasm. It protects it from the activity of the RNA exonuclease. Now, another mode of gene regulation is alternative splicing. Now, there are many eukaryotic genes in which it is not totally specified which nucleotide sequence will act as exon and which will act as intron, or which all sequences together will form one type of protein, and which all sequences of the same transcript will form the another type of protein. Now, which protein is formed depends on the external signals, that is the signals of the external and internal environment. So, as per the need of the hour, see, for example, in this, if these three exons, that is 1, 2, and 3, they join together, they become the part of the final messenger RNA, it will result in one type of protein. However, if only 1, 2, and 4, they become the part of the messenger RNA, they act as exons, and this and other parts, they act as introns. So we will obtain completely another type of protein, that is protein B. So by alternative splicing, also the expression of gene can be regulated in the eukaryotic cells. So these are the major points on which the regulation of gene expression in eukaryotes, they are regulated, and only the genes which are required at a particular phase of life, at a particular time, under particular environmental conditions, they remain active. Thus, optimum use of the resources is made by the cell, the eukaryotic cell. Thank you and all the best.